Maybe not. Janice, can you, okay. So thanks for inviting me to talk today. I'm talking about a site in Sonoma County, north of Sebastopol, if you know the area. A little background about the site. Sonoma County is privileged or cursed to have 18 species that are endemic only to Sonoma County and 70 taxa that are either federally listed or California 1A or 1B. The CNPS local chapter, the Milo Baker chapter, has 4 preserves that we steward to try to preserve as many of those species as we can. This preserve, the Vine Hill Preserve, has 3 appropriately named species. The Arctostaphylos densiflora, the Vine Hill manzanita, Ceanothus foliosus vineatus, the Vine Hill ceanothus, and Clarkia imbricata, the Vine Hill Clarkia. And last fall, the steward manager came to me and said things weren't looking so hot there. Would I come out and take a look? So I did. And I have to say, that's my first involvement in the site in over 20 years. So a little background on the site itself. Vine Hill was purchased by CNPS between 1976 and 1980. Like many endangered plants in Sonoma County, it was these 3 species are endangered due to vineyard development, housing development, and collectors. When the CNPS purchased the property, this is the property in 1976, it had been a vineyard, and for whatever reason, the vineyard was removed and the property put up for sale. Knowing that these few plants up here were the last remaining Arctostaphylos densiflora, CNPS purchased the property, and in 1980 or 80-ish, the our botanist in Sacramento at CNPS office noticed that the manzanitas were looking sickly even then, and asked a nursery manager who was involved in CNPS at the time, took on stewardship of the site. She asked him to start propagating those manzanitas, hoping to get clean material and put out clean plants on the rest of the site. So he, over the next 10 or 15 years, propagated the manzanita and planted them. This is a hillside. This picture was taken at the bottom of the hill looking south. And this is the same view in January when I was out there, looking from the bottom of the hill to the top. These are the remnant densiflora at the top of the hill. And this is some of what I saw when I was there in January. Obviously not happy. This is the manzanita, and this is in January, the last remaining Ceanothus foliosus vineatus. This is some other ones on this site. There are still some fairly healthy looking specimens, but many looked more like this. So I did pair baiting of soil on the site. This is the Google photo looking north, of course. So this is the top of the hill and the remnant naturally occurring manzanitas. And then this is the bottom of the hill where those previous pictures were taken looking this way. So I did one sample at the top of the hill in the natural occurring manzanitas, one in the first swath that were planted halfway down the hill. There's probably a, 50-foot elevation change from the bottom to the top. And then at the very bottom of the hill here, and then a fourth sample taken from underneath a manzanita that was underneath an oak, just to see if it could have been Ramorum.
Betty, I'm just going to give you your 2-minute warning.
Right. And these are pictures of the plants that I took soil from. The first one, number 1 on the map, and the second one midway down the slope at the very bottom, and then the one under the oak. And of course, everybody knows pair baits, 1, 2, 3, 4, and then the results, which when I got to this point, then I packed them up and took them over to Suzanne for diagnosis, and she can take it from there. And I will unshare my screen somehow. Okay, am I off? Is my screen off? Go back. Janice, am I off screen? I can't tell.
You are still there, but I think you should be okay now. I think Suzanne should be able to go next.
Okay.
All right. Good afternoon, everyone. Thanks for the introduction, Janice, and thank you, Betty, for the background information on this super interesting site in Sonoma County. I'm going to pick up where Betty left off and go from there. So yes, Betty said in January of this year, she sent our lab 4 different pair baits. Pair 1 was taken, she said, up the hill towards the road. It was underneath some of the, what we consider the original wild plantings of Arctostaphylos densiflora. And from that pair, we isolated Phytophthora pseudosyringae. So we isolated these onto media, and then we confirmed by PCR. And so this species, it's a root and collar pathogen of many forestry hosts in North America and Europe. It interestingly also causes leaf spot disease. We see it in California nurseries on things like camellia and rhododendron, lauris. And we also see it on bay. People confuse these spots caused by pseudosyringae on, they think it's Ramorum, and it's actually pseudosyringae. Pair 2, we only detected Pythium from that pair. No Phytophthora were detected. It could have been the Pythium lesions just outgrew the Phytophthora. Pairs 3 and 4, which were further down the hill in the outplanted plants, we detected Phytophthora cambivora from both of those, including pair 4 that had the coast live oak around it too. And Phytophthora cambivora, like Phytophthora pseudosyringae, is a forestry pathogen. It causes root and stem cankers on a lot of different hosts, especially members of the Fagaceae family, oak, beech. It also affects alder, maple, and we've seen it. We picked it up in our detections of native plant nurseries, so it is in nursery stock as well. Interesting thing about this species is it's cambivora means cambium destroyer, and that's kind of what it does to the cambium of these hosts. So after we had made these detections, we applied for a California Endangered Species Act plant voucher collecting permit. And this enables you to collect rare and endangered samples. And we wanted to do this the right way because, as Betty stated, there's these 3 special status plants at the preserve. And really, you're not allowed to take any sort of material without a proper permit. So we applied for this permit. And while we waited for the permit, we did make a site visit out to Mine Hill with Betty Young, Sarah Gordon, who's a stewardess there, and Jean Cooley joined us from Department of Fish and Wildlife to look at the site and collect samples from the non-endangered or threatened hosts. So we have a diagnostic permit that allows us to collect from non-threatened or endangered hosts. So we went out in February of 2020. This is looking down the hill right by the road. Immediately you kind of notice that there's a lot of, it looks like there's been a lot of foot traffic coming down the road. And Sarah said, in fact, there's a lot of botanists and people that are interested in seeing these plants. So there's a lot of foot traffic and movement around. We saw some dieback of some madrone from there. There's also Arctostaphylus manzanita at the site. And also there's some hybrid species as well. That's gone on as well. Further down the hill, as you walk down, you start to notice, again, this is the mixture of outplanted, kind of the first outplanting. You can see some symptoms. And then this is more of the area where you see a large amount of mortality among those densiflora. And this really reminded me of, I went out with Ted and Liz from Phytosphere Research a few years ago, and we looked at the Ione manzanitas that were being they're being destroyed by Phytophthora cinnamomum and Ione, and this really struck me as it sure looks very similar to that. Just really devastating to see this on this super endangered species. We also found the Ceanothus, the Vine Hill Ceanothus there. Sarah thought the last two known specimens in the wild, she thought they had died, but we did see them. But they were in super poor health, and it took everything I had not to take a sample from these. Our botanist was with us, and she was making sure I didn't collect a sample, but I was dying to take a sample of these just because I wanted to know if there was a pathogen or if there's a Phytophthora associated with these, because I was worried they were gonna die by the next time we were out there.
Suzanne, 2 minutes.
Thank you. We also saw some coast live oak with some cankers. And so we did some isolations. I thought there might be some remorum. And it turns out these coast live oaks are infected with Phytophthora cinnamomum. And this is a super, super bad Phytophthora, probably most of you know. Has a huge host range, more than 1,000 hosts, forest ornamentals, agricultural crops, and it causes a root and crown rot. And it's really bad in Australia, but we have cases in California where it's doing a lot of damage too. And it's also found in the nursery trade. But these trees are known. These are not. There were no coast live oak planted, so these are natural trees. We saw some foliar symptoms, and I took these as well, thinking there might be remorum. We only detected some Mycosphaeriaceae foliar pathogens, and to me, it didn't really explain the dieback, so it made me wonder what's going on below ground. Same with the manzanita. I took some of these thinking they might be bot canker or remorum, and it turned out to be we only isolated a Cytospora, which is typically a weak pathogen, and it really didn't explain the dieback. So I'm also wondering what's going on below ground here as well. When we were there, Jean Cooley, he kept asking, what about the bear grass? What about the bear grass? And I was so focused on the woody manzanitas and the oaks, and I really wasn't paying much attention to the bear grass. And as we were leaving, Genevieve, our botanist, said, Suzanne, what about the bear grass? And I said, okay, let's look at the bear grass. Well, this is what bear grass should look like. It's Xerophyllum 10X. It's in the Liliaceae family. And these were what they looked like at the site. So they looked really poor. When we tried to dig down, the leaf sheaths were breaking off. The rhizomes were discolored. They looked really bad. And we confirmed that they were infected with Phytophthora cinnamomum from those leaf sheaths. So really surprising kind of for me to see this on this sort of host, but again, it has a very wide host range.
Okay, Suzanne, last slide. Sorry.
Last slide. To summarize, we have 3 species. They're all Pseudosyringae, Cambivorous, and Cinnamomum, all known forest pathogens. We didn't detect remorum. We did find other fungi, but we don't know the pathogenicity of these. We really need to focus more on looking at the roots and rhizospheres of them. We tried to get some funding for biodiversity. CDFA had some money, a pot of money to study this, and we applied for it along with our permits, and lo and behold, COVID hit, and out went all that money and that chance. We do have a permit to collect, but unfortunately we cannot travel during the pandemic. So that's been pretty depressing for us. But we need more sampling and testing to determine the infestation at the site. Are these Arctostaphylos infected with Phytophthora? We really need some tissue, plant tissue to confirm this. We don't know the source, how long these Phytoptheras have been there. And then finally, the special status nature of these flora make it remediation really difficult, especially the Clarkia, because that's federally endangered as well. And so steaming, solarization, those sorts of things might be pretty difficult at this site. So that's all I have.
Thank you, Suzanne, and thank you, Betty. I just want to address some of the comments coming through. It looks like there are some issues with the slide not being fully visible. So we'll work to make sure you can get all of the information that was put out by the presenters. I don't think that'll be a problem. We can share that out separately. And then Suzanne, there was a question, there were two questions, but I'm wondering if you can just kind of consolidate them a little bit. There was a question about visual differences between different Phytoptheras such as Remorum and Pseudosyringae, and this a general kind of question of, is there a way to have a table of visual cues so you can help to identify between different Phytophthora species?
Really, that's a good question. Really difficult. With the Phytophthora Pseudosyringae, the symptoms on the foliage look quite similar between the two species. Same with on the oak as well, you could see cankers causing the same things. We don't really know. The Arctostaphylos, symptoms of Phytophthora that we've seen on Remorum on other Arctostaphylos species tend to be more of a blighty, whereas these symptoms that we're seeing that we're thinking might be a root Phytophthora, maybe Cinnamomum, maybe another root one, they tend to kind of start at the ground level and go up. So it depends on if you're dealing with a foliar Phytophthora or more of a root colonizing Phytophthora.
Thank you so much. I'm going to keep us on time, so I'm going to transition to the next talk. We have Chris Lee with us from CAL FIRE based in Fortuna, and he's going to be giving us a presentation on Phytophthora problems in North Coast wildlands. So Chris, I think you're queued up and ready to go.
Can you hear me now?
Yep, you're good.
Yeah, I don't know why, but it doesn't seem to want to let me unmute.
It's my secret muting power. I think you're live now, Chris, so I think you're okay. If you keep on muting, I'll keep my finger over the trigger to keep you unmuted.
Sorry about that. Good afternoon, everybody, now that we can hear me. And Betty and Suzanne gave some great, if very sad, information there, and I'm going to, in a way, take off from where Suzanne left off by starting with Phytophthora Cinnamomum. My intention is to zoom you through some pictures of damage caused or at least associated with Phytophthora species in the North Coast. And I isolated these Phytophtheras, but they were identified in the California Department of Food and Ag Lab. And thanks to Suzanne and Cheryl Bloomquist and other people at the lab for that work. Also, at the end, I'll show you a map of some places we found it in the North Coast and beyond. And that represents work by Ted Sweeky and Liz Bernhardt and Kim Corella and other people throughout the state. I'm going to confine the pictures to the North Coast for now. So Cinnamomum I've found in several different locations throughout the North Coast. We're going to start up in the upper northwest corner in Del Norte County. It's not a very dramatic picture. If you walk on the ground, it looks more dramatic, and I'll try to get better pictures in the future, but it's affecting a lot of different species in this spot, and there are a lot of potential factors associated with that. As I look through these sites where I'm finding various non-native Phytophthora species in association with plant damage, I've been looking for what other sorts of factors in the soil or factors related to human activity might be tied to this, and I'm finding a variety of different ones. But here at this site, there is historic mining activity on serpentine-derived soils upslope. There's a lot of residential development related activity, including planting ornamentals, stacking madrone firewood, putting pavement and asphalt over tree roots, and there's a generally wet and warm climate in Del Norte County. Moving south to Mendocino County, Phytophthora cinnamomi affects a variety of trees, most of these related to the Fagaceae or the Ericaceae. So here's a site where it's affecting tan oak, rhododendrons, and chinkapins. But there's also a site in Mendocino County that looks more like this one. This one is from Sonoma County, but we have some of these in Mendocino County as well, where it's affecting bishop pine. With the sites in Mendocino County, it is associated with multi-use trails, also with soils with a lot of clay in them, or even with, in some places, very sandy soils that are saturated a good amount of the time. Down here in Sonoma County, there was extensive changes made to the near ocean landscape in the 20th century. Agriculture, plowing, disruption of the soil structure, creating a perched water table. All those things made it perfect for Phytophthora cinnamomi. Here you can see that perched water table. It made it a perfect zone for Phytophthora cinnamomi to proliferate once it was introduced. Now when Phytophthora cinnamomi takes advantage of these trees, other root diseases like armillaria and Viola schweinitzii invade, and they help take the trees down. Now we also have other invasive pathogens in this site, like pitch canker. And here it is in Marin County, affecting a number of those same species. Ted Swicky had already found Phytophthora cinnamomi. This is at Mount Tam. He had already found it in this vicinity, and this year and last year we could see even more damage caused by it, indicating that it's spreading downslope and around the slope. But we've also got some other non-native Phytophtheras that we see around the North Coast. Phytophthora cactorum, which interestingly enough was among a cocktail of species affecting big coast redwoods, as well as tan oaks and madrones here and there. On the right, you can see a picture of a stressed out redwood tree. It was so stressed out that redwood bark beetles were trying to attack it, but it had rallied enough to repulse those bark beetles, which is very cool. Redwood is a very resilient tree, and so even though the crowns were nearly brown, the trees were still sprouting some new green material along the trunk and high up in the crown of the tree. Here's one from Santa Cruz, Santa Clara County line in Mount Madonna, close to Mount Madonna State Park. This was mostly affecting tan oaks and madrones, and it's a Phytophthora that's actually closely related to Phytophthora cactorum. It's actually on the borderline, potentially. It was difficult for everyone to resolve whether this one was cactorum or Pseudotsugi. In other parts of Santa Cruz County, there are other Phytoptheras, Phytophthora cryptogaea, affecting coast redwood. This one's on the bank of a stream. It's near an old orchard. There's home construction nearby. And again, we found other root pathogens present in the tissue of these redwoods. And you can see on the left the healthy redwood crowns in contrast to the ones in the middle. It was also affecting some live oaks to the right. That's easier to see from a drone's eye view than it is from this picture here. But those trees are all suffering this to one degree or another. This one is back in Mendocino County, Phytophthora pseudocryptogaea, killing a large grove of planted Monterey pines. And this has a very high water table here as well. It's right on the ocean. And these Monterey pines are in terrible shape. And the stand seems to be dying from the outside in. again in Mendocino County. And a lot of this in Mendocino County is related to some very interesting fragile soils that we have that coincide with an area of high recreation use and high residential use. This one is a Phytophthora species closely related to Phytophthora cambivora. This is a Monterey pine seedling that was wilting. Another species of root pathogen, a Rhizoctonia species, was also found in this same root system. This is a place where the entire Monterey pine stand has collapsed. Both Phytophthora cactorum and Phytophthora cinnamomi in this site in Santa Cruz, causing heavy damage to a stand of madrones. And this is a place where there was some past logging activity. It's downhill from a road. There's that clay element in the soil and a high water table, all of those things related to that soil type. And then in Del Norte and Trinity counties, there are some that I've just isolated or actually baited from the soil within just the past few weeks. The two on the left from Del Norte County are at the CDFA lab right now. The one on White Alder in Trinity County, I just isolated and I'm just about to send it in. These are all causing varying kinds of damage to varying kinds of hosts. So out of all these examples of Phytophthora in the North Coast, I've tried to extract a few commonalities. Streamside locations or high water tables, they allow Phytophthora to sporulate prolifically. Soils that are nutrient deprived, either because that has effects on the host trees or because there's not as much other microbiota in the soil for Phytophthora to compete with, or for whatever reason, nutrient deprived soils seem to be a likely place to find these Phytophthora species.
Chris, 2 minutes.
Thank you. Soils with a lot of clay, or even some with just a little bit of clay in them, potentially because of the way that the water moves through those soils, or potentially because of the chemistry of those soils brought in by that clay fraction. And of course, as we all know, human activity is a commonality with places where we often find these Phytophtheras. Landscaping, old ag activities such as orchards or plowing, lots of heavily used trails, and so on and so forth. So finally, just to leave you with a couple of interesting points to keep in mind. Sometimes the forest damage that Phytophtheras are causing is hard to see or subtle, and something that I didn't put on here is that it can also be cyclical. There are areas in Mendocino where lots of trees are dying out right now, and they were dying out back in the 70s or 80s, but then they recovered and seemed to be fine until just the last few years. So a good knowledge of that site and how it's looked over the years and decades can help be a clue. And then the potential for damage seems to be very great on human altered soils, or the ones where trees and shrubs perennially just barely hang on in terms of having enough water. And this leads to some interesting questions to me about human uses of our forests. If you have rare or fragile forest types, are those the best suited for more recreation development, or is that going to spread Phytophthora around more? And then what can we learn from the way that they've managed Phytophtheras in other parts of the country and the world? It's a good thing to research and to ponder. And with that, I just have lots of people are working on this and helping out with this, and I extend my appreciation to every one of them. Thanks.
Thank you, Chris. We didn't get any questions for you. We did get a comment from Tyler just saying that characterizing P. cactorum as non-native is not necessarily backed up by the evidence. So
that's a really good point. Thank you. And it's something that I've wondered more about myself, so I want to know more.
Cool. I also wanted to address the question about whether these presentations will be posted to a website. I am recording this just in case we're able to share this for people who weren't able to attend today, and I will be working with the different speakers to see whether we can share their slides. So thank you for that question as well. So now I'm ready to move on to our next speaker, who is Christina Weber with CDFA. So Christina, she's going to be talking to us about the CDFA accreditation to improve restoration program.
Okay. Can you guys hear me? Okay.
Good.
Cool. Hey everyone, my name's Christina. I just wanted to give a brief introduction of who I am since I'm kind of a new face for the BMP program for CDFA. I've been with CDFA since about 2009. I've been doing BMP specific work on and off since 2012. And after Kathy Costa retired, I took over her program almost a year ago now. So the purpose of this presentation is to give you an update, a little background on the program, and an update of where it is leading in the future. All right. So we have traditionally with this program collaborated with NORSDUC, the National Ornamental Research Site at Dominican University. In 2005, the search for the site, which is now at Dominican, commenced at the recommendation of Drs. Nick Grunwald and Jennifer Park, and they proposed to have a project to develop a mock nursery to do research on Phytophthora ramorum. And from everything that I can find, it took some time to find a location, but there was extreme support nationally to get this done, and they eventually were able to find this site. The purpose of NORSDUC, in brief, is to do outreach and research. And the research that's done there, we take that research and the BMPs that are developed there, and we implement them into the CDFA NORSDUC BMP program. And the goal is to share results with nurseries during assessments and audits. The diagnostic services for NORSDUC, another collaboration with our CDFA Plant Pest Diagnostic Center. So that's Suzanne Latham and Cheryl Bloomquist and the crew there. And let's see, this is a neat picture that I found, the ribbon cutting. So you can recognize some faces there. It's been a collaboration since the very beginning. Quick background on the original CDFA BMP manual. It was developed in 2009 with funding from the California Association of Nurseries and Garden Centers. Since then, we've received funding to continue on with the project from the Farm Bill from USDA. And with that, we've done a ton of outreach and site visits to nurseries and just doing educational talks and things of that sort, letting nurseries know that they have resources out there when they have an issue arise. This original manual was designed for growers and interstate shippers of host and associated host plants of P. ramorum. And it was designed to help nurseries take control of any potential problems before they actually arise within a nursery. But several nurseries have been able to use it to get themselves out of the SOD confirmed nursery protocol. Some larger nurseries have been able to utilize it. Oh, I did not know this was going to come up like this. Let me see if I can get it to the end. Nope. Hold on one sec. Sorry about that. I'll just show you like this. So the original BMP manual, like I said, it was a lot of collaboration. So back in 2002, they started, this concept of BMPs for California nurseries. And over a 6-year period, it was vetted through CANGC, through National Plant Board, through other state entities. It's been reviewed by researchers, regulators, and industries in multiple states. And then through that process, in 08 is when we started, 08-09 is when we started the pilot program. So that was just a neat slide that I found that I wanted to show everyone, to show you that it's really been a great collaboration over the years. All right, up to current times. So since 2012, with the detection of Phytophthora tentaculata at a native plant nursery and restoration site, we have been working closely with restoration nurseries. There's been sort of 2 programs that have been going on. There's been the NORSDUC CDFA. We redeveloped our manual to have more specifics about restoration nurseries and how BMPs can apply. And then the U.S. Forest Service has also started administering the NorCal AIR program. Both of these programs are due to the strong desire of restoration nurseries to have a voluntary accreditation program to minimize spread of pathogens into wildlands. So it's 2 programs, 1 goal. And what we've been doing is trying to combine those 2 programs and make it easier for the general public so that there's not 2 different programs floating around. The whole point of BMPs is each implementation of each BMP reduces impacts on businesses, it limits pest spread, and it can offer blanket protection for nurseries. So that's our goal, is to offer protection to nurseries, to give advice.
Christina, you got 2 minutes.
Okay, so currently I'm doing a lot of outreach and education. I'm amending the current manual, the CDFA NORSDUC manual and the Cal Fido's AR manual to make it so that we can all work together and streamline the process. Myself and other inspectors for CDFA are going to be going out and training with the auditors, with Ted and Diana. And then I'm also working a lot on doing rulemaking documents to establish a voluntary regulatory certification program. So that includes program requirements. What do we do if there's a positive? How do we, what are the documentation requirements, things like that. And it'll also include funding. Right now, the programs are funded by grant, the AIR is funded by grant money. But at some point, it'll have to be funded probably through the industry or some other source and that'll help give it stability and ensure longevity of the program. So in the future, right now, I'll start working on website content. So as nurseries participate and they are approved through the program, we'll be able to list them on the website so that, people looking for plants that are for cleaner plants, they can go onto our website and say, hey, these are participants in CDFA's program. We would still utilize our CDFA lab for testing. We would still support research at NORSDUC. And I plan to develop an advisory committee so that that committee would advise the department regarding the program so that we all stay in the loop and we're all working together. So as new research comes through, that can be applied in the program. There will be workshops for nurseries. So, to show them soil steaming, pair baiting, things of that sort. And then way in the future, my thought is, I think it'd be really neat. Right now, we're really focused on water and soil-borne pathogens, but I think at some point, it'd be nice to do like what National Plant Board's done with SINC and be able to start encompassing more and more pests into the program and into the manual. And then wrap up, I think the thing to keep in mind is it's a process, it's a collaboration. If anyone through this process has ideas, we're open to them, and please feel free to get in touch. So this is my contact information. If anyone wants to reach out and is interested in doing research or anything else, just let me know.
Thank you, Christina. We don't have any questions in the chat box right now, so I'm going to go ahead and roll over to our next speaker. Janelle Hillman works for Valley Water, and she is going to be giving a presentation on Phytophthora testing of nursery container plants prior to outplanting in native habitats. So Janelle, you can go ahead and take over.
Yes, hello. So I'm Janelle, and I also want to mention my 2 co-authors for this work, Ted Swicky and Liz Bernhardt. I work in the Santa Clara Valley Water District, now known as Valley Water. And we are a wholesale water supplier for the entire county of Santa Clara. So we supply water as well as providing flood protection for almost 2 million people. And because we have such a large county, we cover quite a large area. And really our projects are located in quite a mix of different types of areas, highly urbanized areas in Silicon Valley, rural South County ranch land in the lower part of our county, and then also relatively pristine foothills. So as a result of our diverse project sites, our mitigation sites are also diverse. Here are pictures of 2 mitigation sites located in very different parts of the county. The first is in downtown San Jose along the Guadalupe River. We have a large homeless population in this area and it's highly urbanized. The second mitigation site is located in the Hamilton Range foothills that are pristine and contain a large block of serpentine soils. And actually, it was both of these sites where we first discovered Phytophthora in 2014. And subsequently, we launched a large-scale testing project of a sample of our mitigation sites across the county. This was done in collaboration with the Rizzo Lab at UC Davis and with Phytosphere Research. And the results of that study, we sampled about 41 different mitigation sites. We documented 50 Phytophthora taxa from terrestrial samples and an additional 6 taxa from water samples. We had multiple detections at a single site, and this included Tentaculata and several locations. So as a result, we've developed both short and long-term responses to this emerging issue, ranging from the development of the MPs and contract specifications to joining the Phytophthora Regional Working Group. And initially, we actually ceased mitigation planting altogether, and we asked for a 1-year grace period from our mitigation commitments from our regulators. We tried planting from seed, but we really soon realized that it was not feasible, nor was it really desirable to avoid using container plants. So this brings me to the topic of today's presentation, which is to give you an introduction to our nursery container stock testing program. So the purpose of this program is to conduct batch level testing of leachate from nursery plants that we are growing for our mitigation sites at various nurseries prior to planting. And in this picture, you can see groups of container plants. They are grouped into batches, and what we do is we irrigate these plants, and then there's a catchment system that contains the effluent or leachate into these blue containers, and then that leachate is baited with pairs to look for potential Phytophthora pathogens. So we have a 3-year contract with Phytosphere Research to conduct the testing, and all tests are run at the nursery with Phytosphere oversight and assistance by Valley Water and nursery staff. The testing of our container stock occurs no less than 2 weeks before the plant delivery date. And typically our testing occurs between September and March because this is our planting season. We can't test during bad weather or at low temperatures. Certain species of Phytophthora are not detectable at lower temperatures, or on plant material that hasn't achieved a specific growth state, being large enough to basically give back a result of Phytophthora. So I keep track of plant orders and contracts for various projects that we have at Valley Water. And this includes the name of the nursery, the project the plants are being grown for, the expected delivery date, the plant species within the order, the number of containers, as well as the size of the containers. This information goes into a master spreadsheet, and then these spreadsheets are organized on testing day by container size and species, as well as the delivery date. The data are used to group the containers into batches that are then tested at the nursery, and then this is the testing plan for that day. If the nursery has its own equipment, this equipment can be set in advance and the containers can be moved into testing the morning of testing.
2 more minutes, Janelle.
So each test requires about an hour and a half to run, excluding the setup and processing. We can run about 12 tests in a day, but sometimes as many as 22 tests in a day if we have additional staff assisting. And we get results after about 8 days if there's no detection, or there's an additional time period if there's a positive detection and samples need to be sent in for DNA sequencing. So the results for our last season of planting, 2019-2020, we had 10 total testing days at 3 nurseries. We tested 3,500 container plants. We ran 121 tests, and this cost us $25,000 for these 10 testing days. And most importantly, we had no positive detections for Phytophthora, although we did have Pythium noted in 15 of the tests. So to conclude, testing is labor intensive. It's not cheap, but it's a lot cheaper than remediation, which has been quite expensive for us depending on the site. We do feel that a robust testing program is worth the cost. And testing prior to outplanting gives a peace of mind. Our mitigation success is extremely important. These are permit conditions and we cannot have our mitigation sites fail. Testing can also help reinforce phytosanitary practices at the nursery. And after 3 years testing thousands of plants, we've only seen 3 positive hits, one of which was in non-heat treated soil, and all of the positive hits occurred in situations where we didn't have full compliance with our nursery BMPs. So in areas where we had full compliance with the BMPs in the nursery, we haven't seen any positive detections at all. And I just would like to thank the co-authors of my presentation, Ted and Liz at Phytosphere.
Thank you so much, Janelle. You actually had a fair number of people with questions. So I'm gonna just choose the first one that came in that was not already addressed by Tyler a little bit, but this question was about whether you can provide the contract language as it involves Phytophthora. Is that something you'd be willing to share with other restoration managers?
Is that the contract language for the BMPs? Or I'm not quite clear on the question. Is that the contract language for the actual testing?
That's, yeah, Cody was the one who asked that for MidPen, and so he was saying yes.
Yeah, so I can definitely do that.
Okay, cool. And you have a couple of other questions, but I'm going to save those for the end when we have a longer time to discuss. So thank you, Janelle. I'm going to now let our next speaker take over. That's Vernon Huffman. He is from the NORSDUC, which you already heard about from Christina, so the National Ornamental Research Site at Dominican University. And Vernon's going to be giving us an overview of soil steaming, treatments, and availability. Thanks, Vernon.
Are you guys hearing me? I can't tell if you're hearing me.
You're okay, Vernon, go ahead.
Thank you. So we're talking about treating soils for planting mixtures in nurseries today, and we're also talking about decontaminating soil for nursery sites. So the challenge that we had to contemplate recently was treating large amounts of potting mix and decreasing the time and fuel it would take to do so. So what we had to consider was improving our technique from a top-down approach to a bottom-up approach utilizing a manifold. So we previously have been steaming soil with this top-down approach that you see here, utilizing the SF20 steamer, a supply line, and a steam sock. And then that steam sock is covered with this tarp and secured with water bags. And it works, but it takes an enormous amount of time. We then started to talk about this manifold opportunity here. And so I began trials of a manifold system treating soil from the bottom up versus the current system of top down. And so we had a direct comparison of the two methods. We went ahead and steamed that pile you just saw that had no manifold underneath. And you can see that after 10 hours of steaming, when I turned the machine off, 7 hours that is, when I turned the machine off, I only had achieved temperatures on 2 out of my 4 probes, 50 C being the target temperature in this case. And the 2 temperature probes at the bottom of the pile had not really moved past 20 C, which is a problem. Same issue, we went over to we are partnering with Watershed Nursery over in Point Richmond and went to steam a 15-yard soil, cubic yard soil from top-down approach. And we did achieve our temperatures, but via this graph, you'll see that our bottom probes didn't reach temperature until 27 hours of steaming. And that was steaming all the way through the night. So again, incredibly time and diesel consuming. Then we worked with Watershed. Well, I had already demoed one here at Norsduck, but Watershed wanted to also improve their method. So we worked with them to go ahead and do a real manifold at a nursery. So this is us building this manifold over at Watershed. And these are 1/8 inch holes that are going to be, this thing will get flipped, and these this steam will come out of the bottom of the unit in just a minute after this is all flipped. There's a formula for how many holes you can drill per foot, per diameter of pipe, per the size of steam unit. And that formula is used and available. That's your completed manifold. This will go in the storage bin area after it's flipped over. And then we have a coupler system. I'll talk about, we do this service for nurseries off-site on a per nursery basis. There's this coupler we use, and we ask the nursery to supply the, that's the end that they supply. This is the manifold in action now. We have it at, this is 15 yards of soil dumped on the manifold. The manifold is suspended off the ground just a little bit to protect the holes, and these are cinder blocks used in this case. Now we have the tarp on top of that soil. This is Wheeler Zamoroni mix number 1 that we used both at Watershed and for my trials here at Norsduck. We don't do any guessing. These are temperature probes inserted into the pile at a grid pattern. These are our temperatures up and achieved. And those are 8 probes there, so no guesswork here. And then here's our results. So back at Norsduck, we put the manifold under the pile, covered the pile, secured the pile, 7.5 cubic yard pile. We reached 50 C in 1.5 hours. So remember, 7 hours, we couldn't even reach our temperatures. Now we've reached our target temp and more in 1.5 hours. Here's our 15-yard mix at Watershed. Now we've got the manifold under the pile, and we've reached our target temps within 3 hours time. And remember, that was compared to 27 hours without the manifold.
Burn in 2 more minutes.
Okay, so we do have a steaming on the go program, and because very few steaming services exist out there, we are going to take our unit and gear off-site here and treat nurseries, and I'll show you a couple examples of that. So here we have a nursery in the East Bay who wanted to they didn't have a known pathogen here. They just had a whole bunch of old material that they had been using for years and a gravel site that they felt they were going to repurpose this site, and they felt they wanted to decontaminate it just in case. So that's what we did. We came over with the unit and put probes 6 inches deep into this material and in some of the pots, and we brought it up. I think in this case, we brought it up to 60 C. That's basically the whole thing and me and what it looks like when we're all set up. And this is another site, unnamed nursery. They had a known pathogen, Phytophthora remorum, out here in this area here. So we went down, and it took 2 steaming events. I divided this area in half. And then you see here my steam setup in action. We have temperature probes buried 6 inches deep throughout a grid pattern on this area. Here you can see my temperature probe setup. I've got 12 probes in this case in the ground. Once things get really hot and really moving, you can see how this tarp expands and bubbles up, and it's pretty impressive. And in a nutshell, this is what we offer on a case-by-case basis off-site. And that is pretty much it, and I can field questions.
Thanks, Vernon. We've got one question which is, how much faster do you think the manifold would get wood chips to temperature?
I think that it would be the same concept. I think that we could do a trial. We could do an A and B, and you would probably see a dramatic, I would say the same ratio would apply. You would see it drop by probably tenfold in the amount of time. Wood chips, though, are more porous, so we are going to get better distribution of heat from the top down and quicker. But again, the premise is still the same. The heat is going to, heat rises, so we're going to speed up the process with the manifold.
That's great. Thank you so much. So with that was the last one of our official presentations. And I would like to just move into more of a discussion section where we can answer some of the questions that did not get asked before. So I also wanted to make sure that if you are ready to leave, we've been on for an hour. I know that's probably the max that most people would want to be online. Feel free to sign off. I will just tell you a couple of thank yous to all of our presenters and for the participants who've been sticking around. I've been really enjoying the chats that have been going on with the questions and some of the answers that have been happening. And I also wanted to let you know, as we round out the last bit of this meeting, I will be sending out a follow-up email with an evaluation link. So you can tell us how we did. And one of the things that will be included in that evaluation is an opportunity for you to share feedback about the work group in general. We have been nominated by the Department of Pesticide Regulation for 2020 IPM Achievement Award. And we are gathering our documents together for that nomination. And if you would like to share anything about what the work group has done for you and your work, we'd love to hear that to put together with our packet. So that will be included in the link that gets sent out to you in the email. But moving past that, let's get back out into some of these questions that we didn't get a chance to answer. So I'm just going to read off a couple of different questions as the speakers come, and I'm going to unmute them. And actually, I'm going to ask Bonnie, will you unmute all of the speakers for me so that I can read the questions? That would be great. So going back to some of the questions that Janelle didn't get a chance to answer, there was a question about where along the Guadalupe the Phytophthora was detected and whether it was near downtown San Jose. And I know that somebody also, I think Tyler, suggested that he had detected multiple species of Phytophthora. So did you want to talk at all, Janelle, about where you had been finding the Phytophthora?
Can you hear me now?
Yeah, you're good.
Great. So I would say, actually, unfortunately, many sites along the Guadalupe River, we have detected Phytophthora. The picture that we showed you was along Reach 6, but Reach 10B has a lot of Phytophthora species. I would say in all of the 41 different mitigation sites that we sampled, the Guadalupe River, unfortunately, had the highest abundance of Phytophthora, I would say higher than any of our other creeks. And I think it's because it's so urbanized and it's easily accessible by many different people in downtown. So it does tend to be an area where we have quite a few different taxa.
Okay. Can you also answer the question about what nursery you use? Do you have your own or several? Just curious, people are curious about where you're getting your plants from.
Yeah, so we use 4 Bay Area nurseries. They're all located in the South Bay, and we're open to any nursery that wants to contract grow for us that will follow our BMPs, which are basically the Phytophthora Working Group nursery BMPs. Currently, we're using 4 nurseries in the Bay Area.
Great. And then one last question for you, Janelle. Andrea asked, does this, what you do with the testing, how does it mesh or combine with pre-planting testing at potential mitigation sites?
We don't test our mitigation sites themselves. We tend to have a pretty good understanding of where we might, I'm assuming the question is about background levels of contamination in the mitigation site.
I'll let Andrea answer that question, I guess, if she has more to put in. Yes.
So we have a mix of sites, as you saw in the presentation. Some are highly urbanized, some are quite pristine. Our pristine sites typically have no Phytophthora. Ted actually and Liz did a study within Santa Clara County of some of the higher watershed sites that are located in open space areas and did not find any background levels of Phytophthora in the majority of those sites. So we feel those sites are pretty Phytophthora free. Our urban sites that are located lower in the watershed along the valley floor, urbanized creeks, do tend to have Phytophthora. We don't specifically test a mitigation site before we plant, but that doesn't lower our level of commitment to both growing clean plants and installing them using clean planting techniques. Our hope is that we're not introducing any new Phytophthora species to these areas, even if there is a background level of contamination.
Okay, great. Thank you so much, Janelle. I'm going to ask Vernon a question now. In your talk, you had said something about a nursery site that had Phytophthora morum. And did you say how deep in the soil you measured the temperature?
Yeah, we deep buried our temperature probes to 15 centimeters or approximately 6 inches throughout a grid pattern.
Okay, great. Question for Christina. So Christina, will your NorCal AIR program return to evaluating nurseries once the COVID-19 pandemic is done? And what can a nursery manager expect moving forward?
Yeah, we can, it would depend highly on your local regulations as far as what COVID-19 is allowing. But there is the potential that if there's interest in people just wanting advice, we could work something out as long as we're following the guidelines. And then for NorCal AR specifically, I'm working with Ted and Diana right now to go out and do audits on nurseries as we can, as things come up and as we're allowed to do so. And then let's see, what can nursery managers expect? So this next year will be a lot of CDFA. We'll be following and shadowing how the program has been run currently, and then see how we have to make adjustments from there as far as it eventually becoming a regulatory program. So this next year, it'll be a pilot run and shadowed as you guys have been running it.
Okay, this is a question maybe also for you, Christina, but also potentially for us as a work group. Is there any centralized place or person that's collecting BMP related education and materials to help train nursery staff and volunteers? Can you talk about what you have available through CDFA?
Yeah, so there is, what I usually use before when I was going out to nurseries is we'll use our manual, and that's usually a really good starting point for people to start to understand what the expectations are of being a participant in the program. And then in general, there are a ton of great websites out there. I think you guys can help speak to that. But at some point with the website production for CDFA, I do intend on having those resources readily available for folks. But right now, yes, there are different entities that have good information. But do you guys have a list of any of them anywhere yet?
We try to put anything that our work group has created up on our calphytos.org website. And if there are specific training materials that you're looking for, please email me so that I can get those to the people who can help create them. And we will try to give you what you need to get your training done. Okay, here's a question. It looks like this may be out to the whole group. So I'm gonna ask this question and then you can chat, anybody in the audience. Has anyone found a good source to purchase rubbing alcohol? And is there a substitute to use? So I'm not sure if any of our speakers have that down, but I would encourage anyone in the audience who knows that to go ahead and chat so we can see it. But I do have a question for Chris Lee. Did you see any forest stand level effects of Phytophthora infestation that would significantly degrade northern spotted owl habitat? And would Phytophthora infected forests be more susceptible to large wildfires that also threaten northern spotted owl?
Yeah, that's an interesting question. I haven't really thought about from that angle. I can approach it from a little bit of a tangent. Someone did ask me the other day if the bishop pine forests that are infested with Phytophthora cinnamomi along the Sonoma coast had characteristics that would make them appropriate for marbled murrelet nesting. And what they've seen from bishop pines up here in Humboldt County is that they believe that some of those trees probably do. So I do think it's a possibility that Phytophthora cinnamomi has degraded that habitat for marbled murrelets. Also, if I continue to see more problems caused by Phytophthora, or where Phytophthora are associated with damage to coast redwoods, then I will start to think that, you know, it's possible that there could be some significant habitat degradation related to that. We already know there's a lot of other wildlife problems related to other Phytophtheras, especially the aerially spreading ones like Phytophthora ramorum. So I would say that there is a possibility there. And some wildfire-related research has been done on fire and Phytophthora ramorum as well. That suggests that when you do have a lot of mortality, depending on the host tree species, there can be significant damage to the soil, and it also can have cascading effects that related to fuels, but also related to forest stand structure that could alter the fire environment in the future. So there's a lot of different possible answers, a lot of different avenues that need to be checked out and tracked down. But I think that there are all sorts of possibilities depending on the tree species and the particular Phytophthora.
Thank you so much. And I want to thank Cody and Tyler for putting in additional references in the chat box while Chris was talking. And also to Ted putting in information about where to find rubbing alcohol or substitutes, as well as Mia and Betty. So you guys are just a great resource amongst yourselves. At this point, we don't have any other questions coming in. So I'm going to call the meeting to an end. I do want to thank everybody again for participating. I'm happy that we were able to see each other virtually, even though we weren't able to be in person. Please keep your eye on your email for additional information from the Phytophthora Working Group. And with that, I will say goodbye. Thank you so much.
Thank you.
Thank you.